Brain Atlas Imaging, an imaging revolution. What is the topic? What is brain atlas imaging? A common challenge faced by neuroscientists is collating the vast amount of information regarding the brain. Taken from multiple imaging modalities and numerous specimens, one solution to this issue, which aims to store and integrate this data, as well as make the information in a logical manner, is a brain atlas. Modern imaging techniques, such as MRI, PET and LFP, have allowed the construction of 3D images, which are composed of serial sections along the coronal, sagittal and transverse anatomical planes of the brain. With an increasing database of knowledge regarding neuroscience, brain mapping can apply this information to a 3D model to visually assign specific areas of the brain with properties such as gene expression, linked pathologies, and forming the basis of the brain atlas. Currently, brain atlases exist not only for human brains, but that of mice, macaws, flies, and pigs. The notable brain atlases include the Allen Brain Atlas, Brain Maps, and Big Brain. Why is this important? Brain atlases hold a great deal of importance with respect to the future of neuroscience. The challenge of conveniently storing and collating information, as well as effectively distributing this information at call, is noticed by many neuroscientists. Brain atlases are one of the solutions to this issue, and by overcoming the problem, they will produce a range of benefits for the scientific community. The most notable is that by providing scientists the ability to share vast amounts of information in a logical format, they can not only design better experiments, but also compare their experimental data with the others' experiments to draw insights. For example, data recorded from an experiment conducted on a group of individuals with a specific neurological disorder can be compared with data recorded from individuals with the healthy brain. Both sets of data can then be comprehensively compared in a logical format on a brain atlas. This allows analysis on subtle differences between the brain that might be connected to disorder. This will lead to improved capacity to diagnose, treat, and prevent neurological disorders. Additionally, not only may this help to identify new ways to combat disease, but it may also be invaluable as a teaching resource. Due to the brain atlases being able to visually display complex information across the brain through an interactive platform, educators will be able to harness this to teach university students numerous scientific concepts. Consequently, the importance of brain atlases in the future development in neuroscience is significant and important. What are the pros and cons? While mapping of the brain is no new concept, the increasing demand on accessibility and functional data has only increased. Modern brain atlases techniques look to redress inconsistencies inherent to past brain cartographies. A major flaw and criticism of early models is the lack of comparability that stem from absence of common spatial reference amongst drawings due to inconsistencies in visual observation. This is a far cry in stereotaxic information that can be gathered correctly. The improvement in structural imaging and implementation of PET and fMRI has provided needed accuracy to mapping abilities and provides a better view of histological examination. The data collected from these scans is accurate to compute 3D models, providing an interface that can better detect structural and functional relationships of anatomical regions of the brain, allowing the scale the size of anatomical figures and features in one dataset, allowing generalizations to be made. The superior ability for brain atlases to combine multiple imaging modalities is also paired with its ability to combine population datasets. Variation between sample brains has been so great that no generalization or localization and function can be made. However, population datasets are obtained and an ideal brain from the group can be formed, allowing examiners to detect common anatomical variation within a set and assistance of complex encoded protocols that are used in modern 3D atlases. The trends detected will not be obtained from the individual scans. A major consideration that population datasets look to overcome is a known issue of concordance among pastellation between structures. Different parcellations of structures often use different parameters in attaining this structure. This can lead to skewed results when the data is later analysed and mapped. In conclusion, the development of modern-day brain atlases is a reality made possible by advances in structural imaging technologies, complex analytical software, and digitized user databases and interfaces. As our neurological cartographic capabilities increase, all three key areas and facets to brain atlas production will need to be monitored, innovated, and assessed. The possibilities for brain atlases will need to look to accommodate thousands of datasets from hundreds and will need to overcome inherent issues with past-later concordance to reach excellence in diagnostic capabilities. Brain mapping has come a long way since Brodmann's drawings, and no doubt has further to go. Thank you for watching.